Hello and welcome to Pharma Television News Review here at Oz Biotech in Melbourne in October 2010. On this show, I have Andrew Bray, who is CEO of a company called BroadVector, which is based here in Melbourne. Welcome to the show.
Thank you, Fintan.
Tell us about BroadVector, because it's a recently emerged company. It's come out of several companies which have now merged. Just tell us a little bit about that history.
Yes, okay, that's correct. What we have is, for a start, BroadVector is a company that's focused on diseases of the ageing population. So that's quite an intentional set of targets that we've looked at. The two therapies that we're concerned with are prostate cancer, early stage prostate cancer, and another condition that also afflicts older people is aseptic loosening of prosthetic hips.
Right. Now, behind every good therapeutic focus, there's an underlying technology. So tell us about what the underlying technology for each of those areas of focus.
Okay, the technologies for these two programs actually come from different sources, but they're very similar. So the prostate cancer therapy technology comes from CSIRO. It was developed by Dr Jerry Both, who is now our chief scientist with BroadVector. And essentially that's a technology that's concerned with introducing a gene into the target tissue, in this case prostate with cancerous prostate. The gene encodes an enzyme which will convert a prodrug into an active drug in the target tissue itself. So in other words, you're generating an active compound directly in the tissue that requires the treatment. In the case of the aseptic loosening, the technology has actually come out of Europe. It's come out of the UK and the Netherlands, but it's the same sort of technology in that a vector of a modified virus, which is non-replicating, carries in a gene which once again encodes an enzyme which converts a prodrug into an active drug in the target tissue. In this case, the target tissue is this loosening tissue that's connected with aseptic loosening. I'll go into that in a bit more detail.
Right. Well, let's go back to the prostate cancer first of all. Tell us a little bit more about the prodrug and how that was discovered and how you see that being applied specifically to prostate cancer.
Sure. The enzyme that's encoded is an E. coli PNP enzyme, and that was first researched actually in the United States, and that's controlled by a company called PNP Therapeutics that's in Alabama, Birmingham, Alabama.
Sure.
And essentially that's that. So the research that was undertaken at CSIRO is focused on making use of that gene in a therapeutic sense. And so essentially what happens there is there's a vector. This particular vector happens to be a non-human adenovirus. Mm-hmm. It doesn't replicate in humans, so it's acting purely as a container to carry the gene in, if you like. Sure. The gene is under the control of a prostate-specific promoter, and that's also a technology that was developed jointly by CSIRO and Sloan Kettering Institute. Now that intellectual property also comes to us and will be assigned to us as well. The gene converts, essentially what it does, the prodrug is fludarabine. So fludarabine is a nucleoside. It's an off-patent anti-leukemia drug. It's been used in clinics all over the world for many, many years.
So it's an established drug.
Yeah, its safety profile is very well understood.
Right.
So in this particular case, we're not using it in the same way that it would be used to treat leukemia, so you don't have repeated uses. It's essentially a short course of treatment with that prodrug. Well, of course, it's a drug in leukemia, but normally it wouldn't have any impact on prostate cancer. But it's converted into 2-fluoroadenine when it comes in contact with the enzyme. So interestingly, the E. coli PNP enzyme causes this conversion, but mammalian PNP enzyme does not. So hence the selectivity.
Oh, right. Okay. So now you've got a drug that's already recognised. It's been through the regulatory process.
Widely used, yes.
And now you're returning it from a function which has a prodrug function into an active drug that's now going to be specific for prostate cancer.
That's right, and the specificity comes from the fact that the gene is turned on essentially in the prostate. The 2-fluoroadenine itself, Fintan, inhibits DNA and RNA synthesis in the cells in the prostate. So its selectivity is not for cancer, the selectivity is for prostate tissue. And so what happens is 2-fluoroadenine is a small molecule compound, a cell which is producing the enzyme. When the prodrug is taken up, it will be converted into the 2-fluoroadenine. That will basically cause apoptosis of that cell, but it also spread out into neighbouring cells and also cause these surrounding cells to also die. So it's not necessary to have the vector enter every cell of the prostate to have a therapeutic effect. However, it will only turn on in prostate tissue.
And just remind us where that is in the clinical development.
That's the clinical stage of this. It's been approved by the Australian regulator, the TGA, for a phase 1 clinical trial at a major hospital in Sydney. So we have all the regulatory approvals in place, all the preclinical has been completed. So this will be first time in man for this drug. And the clinical trial is an 18-patient trial which will be at St Vincent's Hospital in Sydney. And that's happens to be the largest prostate cancer clinic in Australia.
Okay. Let's go to the second program now,
yes.
Because it's for largely for hip replacements, prosthetic implants. Tell us a little bit about that now, because as you said, it's the same principle. You've got a prodrug.
Yeah, this is a really interesting program, and we came across this, of course, looking at a number of projects, and we saw, we came across this really interesting application to destroy a non-cancerous tissue using a very similar technology to the prostate cancer program. Now, in this particular case, the enzyme in this case is a E. coli nitroreductase. The prodrug is a nitroarylazidine, so it's a small molecule compound. Once again, it's unlike the first program. This is not a widely used drug at all. This would be a new drug compound. But once again, this is interesting because we found when we looked into it that there's a lot of people who suffer from this condition called aseptic loosening. So essentially, what happens is you may be aware that the number of hip implants worldwide is growing every year. There's probably about a million hips going every year, year on year. So there's this growing number of people who have hip implants. And by and large, it's a very successful procedure. It's one of the most successful medical interventions, in fact. And you can imagine you're bedridden, you have a prosthetic hip put in, and you're out there and you're playing tennis again. So it works for most people, but over a 10-year period, approximately 10% of people who have a prosthetic hip develop this condition called aseptic loosening. And what that involves is the bone into which the prosthesis is mounted over time becomes replaced by a soft rubbery tissue. It's called synovial-like tissue. And so the joint loses its structural strength. Yeah. And the sufferer loses the ability to walk, and it's also very painful. It causes chronic pain. So purely a man-made disease, because you have to have a prosthetic hip in place to develop this condition. Many people don't suffer from it, but if you suffer from it, the only treatment is to have the entire prosthesis removed, everything cleaned out, and have a new prosthesis put in. So even if the prosthesis is in good condition, it no longer fits, and so it has to be removed. So it's not a case that the prosthesis is broken, it's the body's reaction to the prosthesis.
Sure. Yeah. So obviously the purpose of this therapy is to clean up all that soft tissue.
Yeah, essentially, and what's interesting here, there's been a number of clinical trials performed using this technology, and there was a 12-patient study undertaken in the Netherlands, and the patients who received the higher doses, they showed incredible benefit from the therapy. So what happens there is that the tissue is injected in a single injection into this synovial-like tissue, which basically is between a metal prosthesis and dead bone. So it's highly contained. So the enzyme is in the same way as the other therapy. Over a suitable incubation time of say 2 or 3 days, the enzyme is produced in these cells. The prodrug is then in this case administered in a single injection into that same tissue. A very potent DNA cross-linking agent is generated and it causes cell death. And essentially it liquefies this tissue. So the tissue, which is once rubbery-like, can actually be just flushed out of the joint.
And then you can now replace that with some new cement.
That's one way that it could be used. So, and the interesting thing here, Fintan, is unlike cancer, you could have a great outcome even if you destroy, 70% of the tissue. So you're not trying to destroy necessarily everything, but if you can remove enough of that tissue to allow the prosthesis to be remounted using an orthopaedic cement, then the patient may gain a number of years of additional use of that prosthesis. And if you're in your 70s, there's a large number of people who suffer from this problem who can't undergo this major surgery.
Yeah. So you reduce the amount of time in hospital. There's a health economic benefit.
Oh, absolutely. So you've got two aspects. So there's a group of patients who can't undergo the conventional treatment, which is revision surgery, and then there's others who would prefer to avoid such major surgery.
Sure. So, and that is what stage clinical?
That's completed a number of clinical trials, and we would be taking that into a phase 2A clinical trial, and we'd be doing that in Europe.
And obviously these are all novel therapies. Presumably at one stage you want to partner these programs?
Yeah, that's right. In fact, we have a strong interest in finding pharmaceutical company partners earlier on in the piece. So our business plan in a nutshell is to develop both of these therapies into phase 2, and that may be at the end of phase 2A or somewhere into phase 2B, and then to look to partner, out-license or sell the programs, either together or individually.
Andrew Bray, thank you very much indeed for coming on the show.
Yeah, thank you, Fintan. It's been a pleasure.